Hi, welcome. So, this is the Biology Edexcel Unit 1 May June 2024 paper that I have solved over here, and I will be going through it and just explaining the questions and the answers I have written. This is part 2 of the exam, so it's questions 5 to 8. So, yeah, let's start. Hopefully, what I've tried to do here is I've tried my best to write simple and easy to understand answers that are complete answers, so that you don't need to look at the marking scheme and waste your time looking at the marking scheme and reading from the marking scheme and then having to create your own answer. So yeah, let's start. So since this is the second part of the exam, we have two important questions, which is the asterisk question, which are included in these exams. And I have written beautiful, complete model answers for them. And I will, share a few information, share a few tips on how to answer these kind of questions with you and how to gain most of the marks out of those 6 marks that you can. So yeah, let's start. Now we have question 5. Hemophilia is a group of rare conditions that affects the ability of the blood to clot. Okay, right, ability of blood to clot. These conditions are caused by mutations in the genes coding for factors involved in the blood clotting process. Part 8. Hemophilia is inherited in a similar way to red-green color blindness, and most people who have it are male. Explain why most people who have hemophilia are male. When you're reading a question, it's important that you read the question properly, slowly, you understand and you underline the important parts. So, hemophilia is a sex-linked disorder. Therefore, the gene for hemophilia is located on the X chromosome. Males with the recessive allele will have hemophilia. However, females need to be homozygous recessive in order to have hemophilia. Just to remind you, what is homozygous recessive? Homozygous means the same, okay? 2 of the same gene. Same gene, same allele, okay? So, if for example we have H for not having, not not carrying hemophilia, and H is carrying hemophilia, a woman or a female would need to have HH as her alleles to have hemophilia. Yeah, let's continue now. So we have 2 types of hemophilia, A and B. In 2021, there were 7.8 times 10 to the power 9 people in the world. There were 185,218 people with hemophilia A and 37,998 people with hemophilia B. Calculate the ratio of people with A to B, 2 significant figures. Now here, what I've done, here's A to B. I've written it down so that it's simple and easy, so that we're not making any mistakes in calculating, right? And then to find the ratio, what you're going to do is you're going to divide A over B. And you get 4.87. Now remember, they asked for 2 significant figures. Therefore, our answer is going to be 4.9 to 1. So, which is the percentage of the world population who had hemophilia A in 2021? So, we had 185,218 people who had hemophilia A. So basically, we're going to divide the number of people who had hemophilia A divided by the total world population, and we're going to multiply it by 100 since they asked us about the percentage. Therefore, we get the answer to be C. So, the number of people with hemophilia is only an estimate. Give one reason why this is only an estimate. There are many reasons that you can write over here as an answer. I've written some. So we can say that people are dying and being born within each second. Not everyone has access to getting tested for hemophilia A, or it is difficult to record all the cases. I'm sorry. It was supposed to be cases, not causes. It's difficult to record all the cases. It's not easy keeping track of each person that's being born, that's dying. So there will be some inconsistencies within the data that we have. Moving on, in hemophilia, activation of the blood clotting pathway results in less thrombin being produced than in healthy individuals. Explain how reduced thrombin will affect blood clotting. So we know that thrombin is an enzyme and it catalyzes the conversion of fibrinogen to fibrin. So without thrombin, this conversion occurs, but it occurs at a very slow rate. Therefore, without fibrin, we have fewer platelets that will be trapped, that will be clotted. So if you don't have an enough amount of thrombin, what's going to happen is the conversion of fibrinogen to fibrin will happen, but at a very slow rate. So if someone is bleeding out, if they have like a serious cut or serious injury and they're bleeding out, their body will fill, the platelets will fill to clot at the injured area and stop or reduce the amount of blood that is being poured out. So yeah, this is how a reduced amount of thrombin affects blood clotting and the speed of blood clotting. Okay, now we have question 6. Glucose is an example of a monosaccharide. So monosaccharides join together to form disaccharides, oligosaccharides, and polysaccharides. It's given here that oligosaccharides contain from 3 to 10 monosaccharides. So the diagram shows 2 glucose molecules. Complete the diagram to show how these 2 molecules join by glycosidic bond to form 2 products. So what I've done here is I have just, copy pasted the drawing, right? And right over here, I have shown the glycosidic bond. And under here, I have also shown one product, which is H2O. So for these kind of questions, it's important to not only remember your alpha and the beta glucose molecules, but it's also important to remember the bonds which are created between 2 of them, and just you need to practice how to draw the diagrams. It's very simple and easy. Just practice it a little bit. Take a few minutes to keep drawing it again and again and again, and you'll remember it, and it'll be really easy and simple. So now we have the next question. We have starch is a polysaccharide composed of amylose and amylopectin. Digestion of starch begins in the mouth by an enzyme called amylase. So this amylase can break only 1 to 4 glycosidic bonds. So it's also given here that only disaccharides and oligosaccharides are produced during this reaction. So the diagram shows part of an amylopectin molecule. Draw one disaccharide and two different oligosaccharides that could be produced within the following of the digestion of this part of amylopectin by amylase. So my fault, I forgot to color in my diagram. Now, either for the disaccharides, we can have two just normal white glucose molecules, like here, or we can have two molecules right over here that are joined by the 1 to 4 and the 1 to 6 glycosidic bonds. Now, the same thing over here. As we can remember, this is why it's so important to underline information that's given to us in questions, because right over here, it's given to us about oligosaccharides and their composition. So right here, it says that they contain 3 to 10 monosaccharides. Therefore, when it comes to drawing, you know exactly how, or, you have an idea of what you can draw for the exam. Because if you didn't read that part, and if you didn't remember it, you'd be confused and you'd be wasting time trying to understand, and it would just confuse you. So this part over here was the disaccharides. It's either two molecules, two glucose molecules joined by 1 to 4 glycosidic bonds, or it's two of them which are joined by the 1 to 4 and the 1 to 6 glycosidic bonds. Now for the oligosaccharides, you have a few different options, of course. We can either have it as completely just the white molecules, or we can have a mix of, let's say, 2 gray and 1 white molecule, or it can just be colored in different ways, right? Another one that we can have here is these two being gray and the rest being white, this being gray and this one also being gray, this one being gray and this one also being gray. But it's pretty simple. You just need to remember the importance of reading the question properly. Now, they're asking us explain why this amylase is able to only break down 1 to 4 glycosidic bonds. Now, we know that amylase over here is an enzyme, right? Enzyme that breaks down the bond. So what we know about enzymes is that enzymes, they have, of course, this is not their shape, but, let's draw a cute little Pac-Man, right? So this is enzyme. This is my, let me make it better. Let me draw better. 1 second. Nom, nom, nom. So this over here is my little Pac-Man. He's my enzyme. This part over here, as you all know or should know, is the active site of an enzyme. Each enzyme has its own unique and specific active site, which can only fit and can only bind to the enzyme's specific substrate. What is the substrate? The substrate is the piece that will fit right there into the active site, as you can see here. Nom, nom, nom. It's gonna fit right into there, and the reaction will take place. So amylase only binds to 1 to 4 glycosidic bonds, and 1 to 4 glycosidic bonds, they have a specific shape which only binds to the active site of amylase. So, if it was a 1 to 6 glycosidic bond, it wouldn't fit into the active site. Because here, for example, let's say this size of the shape of the 1 to 6 bond is a square. Now, a square is not going to fit in there. Therefore, You just need to mention that amylase only binds to the 1 to 4 glycosidic bonds, and 1 to 4 glycosidic bonds have a specific shape which only binds to the active site of amylase. That was a small revision. Okay, now we have the digestion of carbohydrates is completed in the small intestine. Okay, glucose is then absorbed into the bloodstream. So just how disaccharides and oligosaccharides are broken down in the small intestine. So glycosidic bonds break down by hydrolysis. Different enzymes break down different bonds in different types of sugars. Again, what I explained up above is that for each specific bond, there's going to be a specific enzyme which will break it down, because each specific bond, the substrate, it has a specific shape which will only fit into the specific or the specified enzyme for that substrate. So we know that when bonds break down, they break down through a process known as hydrolysis. That's important to mention. And a different enzyme will break down a different type of sugar. So, yeah, that's the answer for part CI. See, now we have the next one. The absorption of glucose into the bloodstream requires membrane transport proteins. Explain why glucose cannot diffuse into the bloodstream. So we know that glucose is a polar and hydrophilic molecule, therefore it cannot pass through the phospholipid bilayer cell membrane because it is nonpolar. So we know that the phospholipid bilayer is nonpolar layer. It's not going to allow the polar glucose to go in through. Moving on. Statins are used in the prevention and treatment of cardiovascular disease. Red yeast rice, RYR, is a source of food and medicine in Asia. It is produced by the fermentation of rice by yeast. So we have the RYR contains monacolin K, which is similar to a statin. Okay, the graph shows the production of monacolin K during fermentation of rice by yeast. Over here, we've got the concentration of the K, and here we have the time. Now, it asks us to calculate the mean rate of production of the substance in the culture in a 45-day period. So basically, we're going to go over here on the graph. We have 45 here. We're going to move up. This is our value. We are going to draw a line. Not draw a line, but we're just going to, see what value is right over here. This is 320. This is 340. Therefore, it's going to be 340 divided by 45, which gives us 7.5. And it says give the answer to 2 decimal places. Therefore, we're going to round it up, and the answer is 7.56 milligrams. Now we have the asterisk question. The question that makes everyone nervous, everyone scared. I remember when I was doing my exams, I think I'd either start with the asterisk question, or it would be like the second question that I started with, because I wanted to make sure. Actually, no, I wanted to make sure that I read the question first, so that as I was solving the rest of the exam, my brain was thinking about the question and creating points that I could use within this question. Because when we have here an asterisk question, it's extremely important to remember a few things. One is writing your answer in an organized manner. It's important that you have it, that you present it in a neat and organized way, that you don't have much scribbles or, a lot of mistakes happening there. And another important thing is your grammar, your spelling, your capitalization, everything must be really good. So as you can notice in my earlier questions, I don't capitalize my answers that much. It's a bad habit, but I don't really do it that much. However, when you see me solving an answers question, I am capitalizing all of them, and I'm doing my best to always focus on understanding what information I've been given in the question, understanding the graphs, the tables, everything, and doing my best into adding related information about the information given to us, like from our own knowledge, because it is important for us to not just talk about the information that was given to us, but also to relate this to the information and the knowledge that we learned within the syllabus. So let us start with the question now. It's not that difficult. It looks scary, but take a deep breath. It's gonna be okay. We're gonna understand everything really well. So, scientists have studied the effects of RYR on lipid levels. What are lipids? Fats. So, the table shows the results of 4 studies on the effects of eating RYR on lipid levels. This right over here is our table. So, graph 1 shows, what does it show? Let's see what graph 1 shows. Okay, let's just look at the table for a second, actually. We have 4 studies. So this over here is the mass of the RYR. As we can see, the first study gets to 40 grams. Second one, 1,200. Okay. Third one gets 2,400. Okay. Fourth one, we have split into half and we're giving it 1,200 and 2,400. So from here, like, first point that we can see is that there is an inconsistency within the mass of RYR that was given to the people. That's first thing. Second thing is we can see the decrease in the total cholesterol. We have decrease in LDL, and we have the change in HDL cholesterol. Yeah. So here now we have graph 1 shows the effects of eating RYR daily on the LDL cholesterol levels. It also shows the changes in levels of LDL cholesterol when eating RYR was stopped. That's important. The investigation included a control group that did not eat RYR. Okay. We also have a key given to us right over here. So the control is basically the thickest line, which is right over here. This is the thickest line right over here. So therefore, this part over here is the control, and the other two are, one of them is 100 milligrams, other is 200 milligrams. Yeah. Then we have another graph. So graph 2 shows the results of another study where the effect of eating RYR and LDL cholesterol levels was compared with a control group. Okay. So we have the control, we have people eating RYR. What can we notice here? We can notice a lot of overlapping within the error bars. So that's something that's important to remember and to know. So now the question asks us, discuss the limitations of using the results. The results of these studies to draw conclusions about the effects of RYR on lipid levels. When it comes to answering these questions, sometimes what I like to do is I like to give a small heading before each topic that I talk about. I think it like helps, make it look much more presentable and much more neat. So now let's mention up some points about table 1. So the first point I've written down over here is consuming RYR resulted in a decrease in total and LDL cholesterol. Right? Let's go up ahead and see here. We can notice, see? Here, there is a decrease. Actually, let me just erase this off for you so you can look at it and I understand it. So total cholesterol, LDL cholesterol. Let's look at them both. It's decreasing, 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 decreasing. There's also decrease here. Greater decrease, decrease. It's still continuing to decrease. So that's the first point. The first observation that I noticed that I talked about. The second one I mentioned was the doses of the RIR given to the study groups were not consistent. Therefore, valid conclusions cannot be made from the results. Notice how I'm writing full sentences with full grammar, everything. It is extremely important for you within these questions to write complete answers because it gives you a mark as well. The third point we have is the change in HDL was not provided. Therefore, we cannot compare LDL to HDL ratio to view the effect of RIR. Over here, we were given, change in HDL for the first study and the second study. What about the other two? We were given no information. Therefore, we could not compare the HDL and the LDL levels. Therefore, we can't view the effect that RIR has on the lipid levels in an effective manner. And the last point is there is no data about a control group in the first table. It's not clear if the RIR caused the changes. There is another spelling error right over here. You have to be very careful when it comes to these spelling errors, especially in this question. So, since we don't have a control group that we can compare all of these results to, we don't exactly know that RYR was the reason why there was a decrease within these studies. And therefore, again, it invalidates the results that we have. Now, let's talk about the points in graph 1. So the first one is when consuming RYR, the LDL levels did decrease, yes. However, the LDL levels started increasing once RYR was stopped being consumed. Now, this is another observation that was really easy to understand. Over here, we have RYR started, and here we have RYR stopped. We can see here that as we have it started, we're starting from here, and we're ending up over here. We're ending up over here. We're ending up over here. That shows that there is a decrease. However, now here, once we have stopped consuming RYR, which is right over here, we can see that it is increasing. So that's just one observation that we can see. It's honestly easy. In this question, honestly, I think the best thing to do is to just write down whatever you can think of, whatever you can correlate, whatever you can understand, just write it down because it's going to help you out. So the sample size is small and there is no information if external factors were controlled, for example, the age or the sex. So to add on, there's also a large overlapping of the error bars. Therefore, valid conclusions cannot be made. Over here, we can see that the study group was small. And we can see right over here that that there is an overlapping in the error bars again. Therefore, it invalidates the conclusions that we have. And lastly, we have graph 2. It says that consuming RYR shows there's a decrease in LDL. However, there is a large overlapping between the error bars. Therefore, conclusions will not be valid. and there are no information about the doses that were given. Therefore, it's not. Therefore, these were just the limitations that we noticed in all these graphs and tables. Again, overlapping of the error bars over here, overlapping. It invalidates the conclusions that we have. So now let's move on. So treatment with statins can have side effects. Name 2 possible side effects. I've mentioned a few. We have headache, we have dizziness, joint pain, hair loss, acne, loss of appetite. So many more different side effects that are caused by medicine. It's pretty easy. So now they always ask this question. Therefore, it's important for you to have a model answer that you can rely on so that you can memorize and that you can answer the question, right? So this is my model answer. Sorry. Collect a large group of healthy people with no illnesses, for example, minimum of 100 people. Next, control their age, sex, and other external factors so that most and all of these factors are controlled. Give one group a placebo. No, you have 100 people. You're going to divide that into 2 groups. Give one of the groups a placebo and give the other group the monacolin K. And then you monitor any side effects that are mentioned from the placebo and from the people that are being given the substance. Continue this process out for an enough amount of time. You don't want to just have it be too short, but you also don't want to have the side effects analysis go on for too long. So lastly, what we're going to do is we're going to conduct a statistical test on the data collected about the side effects. We can do this either, using a t-test or a z-test. So yeah, this question is asked a few times, not just unit 1, I think unit 2 as well. Maybe later on as well in unit 4. It's important to have a model answer and just memorize it and, answer it so you have all the points that you need. Now let's suggest 2 problems when assessing possible side effects resulting from treatment. So one is that the placebo could have an effect. The placebo can cause some kind of psychological effect on the people, even though they're not actually receiving the K substance. It's a psychological effect that they may feel or think that they are having a headache or they are being dizzy because of the medicine. So that's one factor. Another factor is that some side effects can be extremely rare, right? Everyone is different. Therefore, everyone's going to have different side effects. Some of them are really rare, and some of them, it's not easy to notice them. It's not easy to recognize them, maybe. Another thing is that different people, they may react differently to the drug. Therefore, you're going to have different side effects. You're going to have different reactions to the drug. And lastly, it is not easy to control and eliminate any external factors, for example, like their diet or activity levels. So sometimes it's the side effects that they feel is not due to the substance or medicine that they're taking, but it can also be due to the change in external factors like diet or activity level. I have the last question. So the role of... Okay, now we have over here the final question. We have question 8. So the role of red blood cells... Oh my gosh, wait, I'm so sorry. Okay. Red blood cells is to transport oxygen around the body, right? The oxygen carrying capacity of red blood cells varies from species to species. The oxygen carrying capacity depends partly on the size and the structure of the red blood cell, and their hemoglobin content. So, describe the structure of a hemoglobin molecule. Hemoglobin, we know that it is a globular protein that consists of 4 polypeptides, and it also contains 4 non-protein groups, and an iron ion is attached to each subunit. So that's all you need to write to get the 3 marks. Just memorize all of these 3, and if they ever ask you about hemoglobin, you know the perfect answer. You have memorized the perfect answer to get all the marks. So now we have the second asterisk question, right over here. Oh, they're always really stressful to me, always so stressful. What I'd like to do, again, I think I mentioned this in the last video, if you haven't watched it. What I'd like to do is I always like to read the asterisk questions first. I like to go over through the whole tables and everything before I start solving the exam. And that way I can, get some ideas on what I can write or what points I can write for exam. And sometimes, you know what you can do? You can scroll all the way down on the blank page and you can just write down points. You can write down points like vasodilation. It's like linking information together over here, or just different things, whatever you want. You can write it down over here. And yeah, it'll help you out with your answer so much. So I'm going to give a few tips now about the asterisk question because they are scary. And, every student wants to get as many of these 6 marks as they possibly can. Okay, I'm sorry about that. Let's go down. Okay. You want to get as many points as you can. So now we have the second asterisk question right over here, as you can see. So I'm gonna give you a few tips on how to solve this question. What I used to do when I was given my exams, I used to read the asterisk question, and I used to, underline information, and I used to look over at the tables and the graphs that were given to me. And what I did was I just tried to do my best to get points, basically. And what you can do is you can just go at the end of the paper here onto a blank page, and you can write down the different points that you have gotten. Like here we have vasodilation, for example. Don't mind my handwriting. I actually usually write cursive, but for you guys, I write it normally. But yeah, just come over here, write a few notes, few points. Or if you think you can remember it in your mind, sure, go ahead, remember it in your mind. But whenever you're answering this kind of question, it's extremely important that you keep your answer. You need to keep your answer organized, okay? You need to keep in mind your grammar. You need to have proper grammar, spelling, capitalization, everything, even your handwriting. When you're answering this question, you need to make sure that you are, as you can see over here, everything is beautifully written. It's, it's beautiful. I took my time. It was beautiful. You can see that I have, I don't usually capitalize my letters, but here they're all capital. Over here as well, I also have bullet points that I have added in. It helps me answer much better. So if you think, bullet points or, anything they'll help you write your answers better, go ahead, do that. It's really good and helpful, and it saves time. Yeah. Getting like a full mark in these exams, especially in AS, is not that difficult. Because I was able to get a full mark in, I think it was Unit 2. I got a full mark in my Unit 2 paper. It wasn't that difficult. You just need to understand and really read from the book, And you need to make sure that when you're answering these questions, these asterisk questions, that everything is organized, and before you start actually writing down your answer, you need to like think of your sentence in your mind, and then you need to write that out, because they don't like it when you write something and then you start scribbling on it, or when you like make a mistake. They don't like seeing scribbles. So basically, I'll give you an example. If you have a word over here and you want to get rid of it, for example, I have photosynthesis, and I'm like, ugh, I don't want to write. I didn't mean to write photosynthesis. I basically put brackets and I put a line in between. Have it look nice. Have it look neat. This looks better than you just going like this. So much better. Right? Okay. Yeah, so now let's start with the actual question and let us go through the points that I have written down. So the table shows some information about red blood cells from 4 species of air-breathing animals. The species are listed by decreasing size. Okay. As you can see over here, the size is decreasing as we go down the table. So over here, explain the relationships between, let's see, the size, the characteristics of the red blood cell, and their oxygen carrying capacity. Use information in the table to support your answer. Well, it didn't ask us for our own knowledge, but it's always really good to add stuff from your own knowledge. Okay? Try to, compare different try to just try to join like the information given to you from the table with your own knowledge. So yeah, let's see. The first point I have here is there is no clear correlation between the size of mammals and the mean number of red blood cells. Let's go check that out now. Okay, here we have the size. As you can see, it's going down. And here we have the mean number of red blood cells. Really quickly, what I'm going to do is I'm going to order these values from, from the greatest to lowest, right? The greatest one we have is the greatest value right here, 1. Second greatest here is 2. Why? Because you have to read this properly. It's the power of 13, power 12. And then we have, oh my gosh, sorry, 3 and 4. So this is them ranked in order. Now, as you can see, beluga whale, which is like, the largest one, it's at 3.3 times 10 to the power 12. Now we're moving on to a mammal which is, smaller in size. However, it's increasing. Over here, it's decreasing, and here it's increasing. Let me just write that in red, actually. Here it's increasing. Sorry. Here it is decreasing, and here it is increasing. Now, let's compare this one. Actually, no, sorry. This one to this one. Okay? Here, it is decreasing as well. And here, it's decreasing. So, okay. And then we can compare this one with this one and this value with this value. We can see here it's decreasing, and here it is increasing again. So this shows that there is no clear or no exact correlation between the size and the mean number of red blood cells. And that was the first point that I mentioned over there. Now, the second one, the mean mass of hemoglobin decreases as the size of mammals decreases. Why is this? Because as smaller animals, this is because smaller animals require less energy Therefore, they will respire less. We know that when we respire, we break down glucose to produce, not to produce, but we break down glucose to gain ATP, which is energy. So if we are, it's a smaller animal, it requires less energy, therefore it's not going to respire more. I guess respiration does take energy and it does take time. So yeah, if it's not needed, then it won't take place in the body. Now let's look here again. So mean mass of hemoglobin and the size of mammals. Let's see. Over here, I have the mean mass of hemoglobin. Here I have the size. Okay, let us look at that one. I'll highlight for you guys too. Why not? Why not? So you guys can know which one I'm looking at right now. So here we have the size, and here we have the mean mass of hemoglobin per cell. Awesome. So again, this is the largest one. Largest one has 57. Okay. This is the second largest one. It has 14. Man has 29. Goat has 7. As you can see from here to here, it's decreasing. Let me get the black color for you guys, okay? So from here to here, and from this value to this value, it is decreasing. They're not decreasing at a constant value, but it's still decreasing, right? So we can understand why. Now, again, this part over here, but smaller animals requiring less energy, it's us relating the knowledge that we have learned to the knowledge that was given to us from the table, okay? So now, the third point I have here is mammals with a high number of red blood cells have a lower mean mass of hemoglobin. So high number of red blood cells, lower mean mass of hemoglobin. Now we are comparing I hate it when I underline. It doesn't look nice. Number of RBCs to the mass of hemoglobin. Let's go back up over here. I am looking at the mean mass of hemoglobin and the mean number of red blood cells. We said that high number of red blood cells have lower. Let's see that, okay? So, as we can see, the values over here are inconsistent, right? We have one which is high, one which is high, okay. One which is high, one which is low. So, what I like to do, what I did was I ranked them in order because I don't know, I think it helped me out. So again, we're gonna rank them in order 1. Sorry, this one here is 1, 2, 3, and 4. Again, going from the greatest to lowest. And let's see now. This is the highest value, and over here we only have 7 grams. It's not grams, but 7. I'm not sure what this is. I think it's perigrams, but I'm really not sure. But yeah, 7 grams of hemoglobin per cell. The second highest here, it has a little more. Yeah, 14 grams. Third highest has a little more. And then the least, like the lowest, number of red blood cells we have has the highest number of hemoglobin in it. So yeah, this is true, right? As the value of the mean number of red blood cells increases, the value of hemoglobin per cell decreases. So they're inversely proportional. That's the relationship that they have. Now, the last point, of course, there are some other more points that you need to mention, and I will mention them. But yeah, cell volume decreases as the size of mammals decrease. So again, we're looking here at the cell volume. Size of mammals decreases except for camels as their blood vessels are wider. This is so that camels can get rid of excess heat during temperatures through vasodilation. Again, this is from own knowledge. I'm so sorry. My writing is so sloppy right now. I apologize. But yeah. Let's go ahead and see what's going on. Let's look at the cell volume now, along with the size. It's important to like highlight or underline and see what you're comparing. Because before I was looking at the wrong things, I was looking at the mean cell volume and I was comparing with the mean number of red blood cells, and I just wasn't understanding it. Because when you look here, they're kind of inverse. Not they're actually I think they're both proportionally proportion. I'm sorry. um I forgot the word, but they're both, if one increases, the other one also increases. Directly proportional, yep. I'm a little tired right now. I'm so sorry about that, you guys. But I'm doing my best to have this video out for you guys. Right? Okay. Yeah. So we are looking at the mean cell volume and the size, right? We have again 1, 2, 3, 4. It's descending order. One over here has 134. Second one here 319. Third one has a 90, and fourth one has an 80. We can see that as this one decreases, the values over here also decrease, except for camels. Again, we said that camels were an exception due to the blood vessels being wider, the blood vessels dilating, so that the camels can lose excess heat in their body to their surroundings. This is an adaptation. This is an adaptation of camels. So yeah, this was correct. So this is like also part of the answer. These three factors affect the oxygen carrying capacity, right? These are factors which affect the oxygen carrying capacity of blood. However, there are also there are a few other external factors which also affect oxygen carrying capacity. So it's not just these three, no, there's more. And an example of that is the affinity of a molecule for oxygen. Some molecules have a higher affinity, other ions or molecules, they have lower affinity. So yeah, this also affects the oxygen carrying capacity of the blood. This is all I wrote for this question. So yeah, let's move on. Now part C, we have the graph shows the oxygen dissociation curve for hemoglobin and myoglobin. So these are both, molecules, and they both have a different affinity for oxygen. I'll mention this up later on in one of the answers, right? This is another factor which affects the oxygen carrying capacity of blood. So myoglobin is present where inside muscle cells and acts as an oxygen store. Again, it has a higher affinity than compared to hemoglobin. That's why it stores the oxygen more. And it's found inside muscle cells. Okay. So over here, we can say we have the percentage saturation with oxygen. Here we have the partial pressure. We have one line for myoglobin, other one for hemoglobin. Let us see. So the decrease in percentage saturation with oxygen of myoglobin from 6 kilopascal to 4 is 3%. Now they're asking us to calculate the difference between this decrease for hemoglobin from 6 to 4 kilopascal. So that is very easy. over here we have 6 Pascal, which is right over here. It's between actually hemoglobin and myoglobin. We're comparing both of them, right? And 4 Pascal right over here. Let's just quickly get that. So the first value that we have over here, not the first value, but like here, we have at 96. This one over here is over here at 76. So this was for 6 kilopascal. So here, percentage change at 6 kilopascal was 96 minus 76, which is 20%. And then we have at 4 kilopascal, we have the values at 4 kilopascal. This one over here is 50%. And this over here is 93. Okay. So at 4%, we have 93 minus 50, which gives us 43%. Therefore, our percentage difference will be 43 minus 20, 23%. So the answer is 23%. Yay. Now, they're asking us to describe the effect of the partial pressure of oxygen. Partial pressure of oxygen on the percentage saturation with oxygen of myoglobin. Use values from the graph to support your answer. So, as the partial pressure of oxygen drops from 12 to 1, we can see that the percentage saturation of oxygen decreases slightly, and the partial pressure drops sharply at low levels of oxygen. Let's see. So right over here we can see we have 2 lines, right? We have one for myoglobin, and we've got one for hemoglobin. Now, the question, this second part is asking us about myoglobin. So we're going to look at the myoglobin line. Over here, we can see that it's pretty much a straight line, and then it starts slowly decreasing right over here. So the decrease right over here isn't that great. It's not as great as compared to this decrease in hemoglobin. Because hemoglobin, we can see it's right over here, and then it just rapidly goes all the way down there. It's steep, right? But myoglobin is much more calm, much more slow. And we can see that it starts decreasing more here to here. So again, well, let's have it be more accurate, right? This is a more broader range. Let's shorten the range, as you can see, from, I think, here to here. So yeah, when there is a low, especially when there's a low percentage saturation of oxygen, that's when the partial pressure of oxygen, sharply declined. So yeah, that was the answer. Now they're asking us to just why the dissociation curve for myoglobin is to the left of the curve and not to the right. So as you can see over here, the line hemoglobin, as you can see, it's like more towards the right. And the line for myoglobin right over here, as we can see, actually, wait, let me draw that bigger. See, it's more over here to the left. So they're asking us why it's like this, why the dissociation curve is like this. So what I wrote was that myoglobin has a higher affinity for oxygen than compared to hemoglobin. Therefore, myoglobin will only release oxygen when the partial pressure is low, so that myoglobin can supply respiring muscle cells with the oxygen when it is required. Right? So hemoglobin provides oxygen at all times. But when you're working out and when your muscles are using up oxygen and hemoglobin isn't carrying around enough oxygen, what happens is that the oxygen stored in myoglobin, once your body is using up oxygen, and once the level of oxygen is decreasing within the body, what happens is that myoglobin starts giving out. It starts releasing the oxygen that it has stored so that your body can use it for respiring. So the muscle cells, which are respiring, can use this oxygen. And that is finally the end. Yay! Again, thank you so much for being here, and thank you for watching. I really hope that I explained things to you properly. I hope that you understood everything. I hope it was simple for you to understand. If you want, you can always subscribe. And if you want, you can leave a nice little like. Oops, that's supposed to be a thumbs up. I don't think I can draw with a subscribe, like, all those things. I'm gonna be posting more past papers, and I might start posting some more revision notes for you guys. Anything to help you out, really. But yeah, again, what I aim to do with these videos is I aim to help you guys. I aim to help revise. You know, my gosh, I'm sorry. I aim to do a nice small revision of the topic. I want to refresh your memory. I want to explain things in a simple way. I want to provide you guys with some beautiful, organized, well written answers so that you don't have to spend so much time going to the past, like the marking scheme of the past paper, and trying to write your own answers. Because when I was studying and I was solving past papers, I would spend so much time just going all the way back to the marking scheme and then going all the way back to the books, to the notes, and trying to write an answer that was acceptable, because that's just how I like to practice my past papers. So yeah, these past papers are excellent for last minute revision, for last minute solving past papers, because whenever I'd study, I would always study last minute. So yeah, that was it. That was the whole paper. Thank you so much for being here, and yeah. If you want me to solve a specific paper, leave it down, or even a question, like just a question, leave it down in the comment section. I'll just solve only that question. I'd love to help out, because I know what I know what you're going through. Also, it's Unit 1. It's like really stressful. You're only starting out now. It's your first time doing these exams, and you're like, what's going on? But no, AS is very simple, very easy. And I will say, do your best now. Like, try to aim and get as many marks as you can during your AS, because it definitely gets much more difficult during your A2 to actually gain marks. So yeah, again, I got full UMS marks in my AS. It wasn't that difficult, so you can definitely do it. Trust me. I I was someone that studied and did my A levels at home by myself. I didn't have a tutor. I didn't have nothing. All I relied on was some YouTube videos and just some notes that I found online, and the book. So yeah, I'm here to just like help other students out like me. Again, thank you so much. Yeah, just let me know. If you want me to explain anything, let me know and I'll be here. Okay. Thank you so much. Bye. Take care, huh? Make sure you get enough sleep and you're drinking enough water.